just the rest of this code in a recursive algorithm. So I could write it, and not just go get some code online and just copy and paste.
Take it this semester because it's just like...
With a series of workshops on how to communicate, how to be on stage. Criteria, their communication style, the comprehension of their talk, and their engagement with the judges in our audience. Those are the rubrics our judges are going to use as the students are up here on stage. They have 3 minutes. They're clocked down here. We'll count them down to 3 minutes to where they can see it, and they're allowed one slide to use as an audio visual. I know our students backstage are really excited. I can't wait for you to get to see what we've worked on for the past few months. So without further ado then, I will introduce the MC of our event, Dr. Gordon Reinhardt.
Live applause, can't beat it. So my name is Gordon Reinhardt, and my job is just to kind of keep things moving. I've done this for a few times, and I can assure you you're in for a good time. It's a rare encounter between the innocent and the informed, and I really enjoy it. Let me start by introducing our judges and thanking them for being here. First, of course, is Lauren McLean, our mayor of Boise. Good morning and welcome, Mayor McLean. Lucy Willits, a member of the Boise City Council. Welcome and good morning. Blossom Johnston from the Idaho
powerful than any computer from the 1950s, and none of those computers would have fit in my car. So it took 70 years of computers getting smaller, faster, and cheaper for this to be something that you could mass produce and put into a car and, potentially save my life. Which brings me to my thesis. Here I have two programs for you, A and B. If you ran these programs, you would never be able to tell the difference. They will look exactly the same, but they are different. Program B is actually significantly faster than program A. I'm not going to get into why exactly. It doesn't fit into 3 minutes. I work on programs called compilers. They will look through your code and try to find things that look like A and automatically replace it with things that look like B. It's very difficult to write these. You have to get really sweaty and drink too much coffee and stay up late to manage to do it, because they actually have to prove that for all inputs, these two programs will do the same thing before they can replace it, and they don't have enough time to actually run the program, so you have to do a bunch of fancy math to try to prove this and replace it. It's really hard. And you might ask, if this is so hard, is it worth doing? And I would say, I think it is, because if you take enough stuff like this, put it in a big pile, that's what allows 70 years of computers to get faster and save someone's life when they're dumb enough to not look at the road while driving. Thank you.
So with each participant, we'll chat a little bit, so give the judges a little bit of time to process and take some notes, and allow you to get to know the participants. So, Aaron, you spend a lot of time. You mentioned late night coffee and sweats and hard work in front of your computer screen. What do you do to recreate, to get away from that?
I really like climbing. Boise is a
Climbing
great place for
anything,
that.
or just climbing?
Rocks.
Rocks, okay.
Rocks, yeah.
Good. And do you see any comparisons between that work and the work you do in front of a computer?
Oh, yeah, completely.
Really?
Like, they're both just a very difficult thing that you have to try over and over again. That's very frustrating.
When you're working on writing code, do you think about rock climbing as a metaphor for the work you're doing in front of the computer screen?
Only in so much as they're both frustrating.
I would imagine, which one do you consider more dangerous?
Oh, definitely the rocks. Well, actually, maybe the code.
Ah, that's interesting. Maybe more dangerous for us,
Yeah.
but rock climbing more dangerous for you.
Yeah, I would say so.
Well, Aaron, thank you so much.
Thank you.
Well done. Okay. So, yeah.
This way?
Thank you, sir. Okay. That timing feel about right for you? Okay, good. So, next we have Mr. Jacob Heil. Welcome, Jacob. Okay, Jacob, nice try, but there's no escape.
Try to run.
That's right. So I'm fascinated. You're aware of a whole universe that, of course, we don't have visual access to. Do you see comparisons between that universe and those entities moving about, living their lives, essentially, and this universe?
Absolutely. I think as part of my research, I've realized how much microbial communities act just like plant communities or even animal communities. We use a lot of the same methods to measure them and the same statisticals to understand what they're doing. But I think like some of the main things we study is like how do they arrive? It's called assembly. And so you can think of like a field, and it's probably got birds and mice and foxes and all that kind of stuff. And they all arrived somehow, and there are factors that influence how they came to live in that field together. The same thing's true for microbes. The same thing's true for plants.
Well, it's funny, I'm hearing words like actor, which of course I relate to, and community, and assembly, freedom of assembly. It's interesting that the language kind of echoes our community. Well, last question, what is it you do to get away from the microscope?
I like to go fly fishing. I like the outdoors a lot, but I'd say fly fishing is probably my favorite thing.
When you catch a fish, are you at all disturbed by the micro level of activity within the fish? Or do you just cook it right up and eat it?
I think in my biological education, I had to put away the gross stuff a long time ago and just be able to deal with it.
Understood. All right. Thank you, Aaron. Thank you so much. Well done. We're having a little bit of trouble, and we apologize for some of the technical issues with the live streaming, but we're all over that. So let's move on now to our next participant, Megan Kelly Slatten. Welcome, Megan.
Plants. When I say this word, we all think of drastically different things. Some of you may have imagined the sagebrush out in the desert, and others may have thought of the brightly colored tulips we'll see shortly in the spring. Regardless of the plant that you thought of, they all have something in common, and that's their ability to capture atmospheric CO2 and convert it into stable soil carbon. This unique attribute of plants may just have the ability to help fight climate change. This is what my research focuses on. I use biofuels not only for their alternative energy, but also for their ability to capture atmospheric CO2 and turn it into soil carbon. Now, once this carbon enters into the soil, it doesn't just stay there. It actually interacts with the soil microbial community. In 1 tablespoon of soil, there are millions and billions of microscopic organisms that come along and eat up this carbon. Now, when they eat this carbon, some of it is turned into microbial biomass, and the rest is metabolically respired as CO2 to the atmosphere. Now, in order to increase soil carbon storage, we want more biomass and less CO2 respiration. So how exactly do we do this? Well, it actually comes down to the chemistry of the carbon inputs. And in order to understand this better, we're going to take M&Ms and hunting a deer as an example. When you open a bag of M&Ms, you don't just have one M&M. No, you eat that entire bag of M&Ms and you enjoy it. But unfortunately, there's not much nutritional gain in that, so you're still hungry afterwards. This is similar for microbes. They like to eat the simple carbon first because it quickly grows their biomass. But unfortunately, a ton of that carbon is then respired back out as CO2. This isn't great for soil carbon storage. Now, alternatively, we'll think of hunting a deer. This requires quite a bit of effort. You gotta get your camo on, go in your truck, go to the forest, find the deer, shoot it, take it home, clean it, cook it, and then finally you get to eat it. That's a ton of work, but now you're nice and full and satisfied. This is similar for microbes when they eat complex carbon. It takes them a while to break it down, so their biomass grows extremely slowly, but they respire very little CO2 in the process. Now in reality, we need both M&Ms and deer in our lives to make us both happy and healthy. This is similar for microbes. They need both simple and complex carbon in order to grow to an efficient size with minimal CO2 respiration. This is what my research focuses on. I use biofuels to try to find this balance between simple and complex carbon that leads to the best functioning microbial community that promotes soil carbon stabilization. With this information, land managers and farmers can then select crops or plants that increase soil carbon storage. By increasing soil carbon storage, you not only promote plant health and soil productivity, but you also fight climate change. Thank you.
Thank you, Megan.
Yes.
So, in searching for an analogy to make clear for the innocents in the
Yes.
audience, do you have personal experience with M&Ms and deers?
I do not, actually. I do enjoy M&Ms, but I've never hunted a deer, so
So-
it's a little ironic.
Can you take me into the process of like, so you know, like, okay, here I have to make this point, I have to make this clear
Yes.
to a layman audience. What didn't make the cut? Like, do you have other analogies in mind?
Well, it actually started with fast food and healthy food and the human metabolism, but it took me a while to tone that down, so
I see.
switched over to something a little bit easier.
Well, I think you found the perfect, M&Ms, of course, microbes, M&Ms,
Yes.
very good. Well, thank you for that. So my question that I ask everyone is, what do you do to rejuvenate yourself after all of your research activity?
I prefer the warmer weather because I love to go backpacking in the Sawtooths and Stanley.
Right,
It's
right.
my favorite.
So you don't backpack in the winter?
No, I'm not a cold person at all.
Yeah. No, I get that from you. I mean, anyone that would come up with M&Ms is clearly not a cold
No.
person. Well, Megan, thank you so much.
Thank
Well
you.
done. Appreciate it. Okay, thank you, everyone. Our next participant is Toby Papula. Welcome, Toby.
Hi. So we all pick our phones up and we check the weather. We check a lot of stuff. And what is actually running in the background is computer servers. So computer servers actually answers your request when you ask all those questions from your phone. So you should know that computer servers take as much as 2% of the world's electricity. You should also know that computer servers expend as much CO2 as the airline industry. So what's going on in these computer servers? We have applications running on these servers, and these applications, they take up all this energy. So my research area answers the question of how we can make these applications run faster, work better, and that's what my research is focused on. So you see in these applications, the major thing that takes a lot of energy is data movement. So if you want to go to the grocery store to get something, you're going to spend more time going to a grocery store 2 miles away than going to a grocery store right at the corner store, 0.5 miles away. We all understand that. We're going to spend more gas going to a Walmart 5 miles away than going to a Walmart 1 mile away. That's the same thing that happens in the computer world, data movement and data processing. So my research area, major focus of my research area is how we can make the data as close to where it's been processed as possible. The other thing about my research area is how we can automate this process, how we can make this process hidden in the complexity that people that write these applications don't have to deal with doing it themselves, because it's really complicated. It's hard to do. It's hard to come up with these transformations or come up with this process to make this work. So my research area also focused on how we can automate this process. So what this means is that the same application running on these servers for the same work will spend a lot less time. And my research has shown that you can actually, based on this technique that is a bit complicated to explain, you can actually get as much as 9 times speed up. So just imagine we're spending 9 times less CO2, or we're spending 9 times less power on this computer servers, or we're spending 9 times less money running these servers. And that is what my research focus is on. A lot of us just use phones and we don't understand that this stuff is taking as much power, as much energy, as much CO2 than other things that we focus on that causes climate conditions and climate changes and all of that. So that's what my research area is focused on. Thank you so much for listening.
So Toby, if I understand, you're promising that I will experience 9 times less frustration with my technology if what you say comes true.
Yes.
Excellent. I could use it. My question is, my first question, do you experience frustration with technology?
Actually, I do.
Yeah.
And the thing is, like, the bad part of it is, like, I understand what's going on.
I was going to say, I wonder if my frustration of ignorance equals your frustration of knowledge, or do you think maybe yours is greater because you know what the problem is?
Yeah, I think it's greater for me because I do know what the problem is, and I just like probably try to apply to those places and see if I can get a job there and fix it.
Okay, well, good. Next time, I'm just going to think of Toby when I have a problem with technology and know that, well, he's suffering more than me, so it can't be all that bad. And I appreciate that, Toby.
Yeah, thank
well, all this computing power, what do you do to get away from it?
I usually just make music. It's still computing power to make music. But
You just use it differently.
I just use it differently.
That's fantastic. What kind of music do you like to make?
I just do like general music, like rap. I'm currently going to perform in Treefort. So,
Fabulous.
so if you're Treefort, come to
So
my venue.
with the computing power and your voice. Mm-hmm. Wonderful. Well,
Yeah.
thank you, Toby.
Thank
Well
you
done.
so much.
Bravo. Okay, thank you, everyone. So next we have Randy Wichman. Welcome, Randy.
Thank you. Okay. All right, so we don't usually conceptualize our childhood photos in accordance with the trauma that we experience, but that is why I asked my mom for this picture of me around the age of 6. Now, as a child who grew up on a ranch with both cattle and sheep, to say that I made it to 6 years old without some trauma would be misleading, but this child had not yet begun experiencing unwanted sexual touch within her school setting. I did my thesis in order to conceptualize the way that gender relations function within rural settings. A rural space in my community, Central Montana, is categorized by 2,500 people or less. I graduated with 6 other people, and I spent a lot of my time working through narrative using autoethnography, which is essentially autobiography compared or in communication with current literature on rural spaces, on gender relations within specific cultures. And that's what I spent my time doing, looking at the ways that these traditional spaces where patriarchal families are common, where we presume goodness and security and safety in harmonious, tight-knit spaces, where all of these exist, and yet we are still experiencing violence within these safe spaces, within these communities that have been represented in the media as healthier because outdoor living, better, what do we call it? Better lifestyles, better relationships. Now, it matters because we have these wonderful aspects of rural communities where they are with many social support systems, but we are also looking at populations that do not have ample access to resources like proper medical care. Domestic violence rates are higher in rural communities despite there being less people. And with all of that, we don't have the proper support for these people, for my family, for my friends. When your value and your moral statue within the community relies upon your ability to be productive and offer the community things from your family and from your skill set, coercion becomes a really effective way to get people to organize and behave the way that you want. So making sure that rural populations are appropriately represented in research is extremely important. Thank you.
That was great, Randy. That was great. So it must be a challenge when your research area interweaves with your personal life and the courage to take that on.
It is tricky. Yeah, autoethnography is one of those methods that's incredible for working through traumatic experiences or through very confusing things like having people you trust, trusted adults, be both helpful and hurtful.
Right.
And not being able to use your voice, especially if you're a member of a community that is marginalized.
Right. It strikes me as wonderful that that you would take a question that you need answered, you pursue the answer to that, and then the answer probably helps others. So thank you for that.
Yeah.
It's a challenge, I'm sure, emotionally and physically, but what do you do to mitigate that as a scholar? What do you do to get away?
Oh, get away? You get away from your research ever? Couldn't be me. However, I would say, like, with this research, it's very important to engage with your community and your support systems.
Right.
And being able to talk about it with people is very important, because that's how trauma happens, is
Yes.
not being able to discuss these things. So
Right.
getting away from it is like engaging with it and then stepping back and then having those support systems that do fun things with you. Go on
Yeah.
a hike. I don't know, knit, things like that.
Go on a hike and knit after you've gone on a hike.
Well, at the same time, whatever.
That could be dangerous.
Yeah, well,
Yeah.
you've got to watch where
Right.
you're going, for sure.
Well, thank you so much and well done. I really appreciate it.
Thank you.
Yes, you're welcome. Thank you, Randy. So next we have, our next participant is Abu Saeed Chowdhury. Welcome, Abu.
And effective communication is the key to our survival. Bacteria also communicate among themselves via chemical messages in an environment like in the lungs to count their population density. Having a minimal threshold level of population density is the prerequisite to regulate their group behavior. For example, turning them into an infectious mode from a non-infectious mode. This phenomenon is known as quorum sensing. Pseudomonas aeruginosa bacteria causes pneumonia in immunocompromised patients, follows this mechanism. Now, these chemical messages are very bacterial species specific. For example, if we have 3 different bacteria, blue, green, and pink bacteria, the blue bacteria only produces and responds only to the blue signals, not to the green or pink signals. Now, how does the signals are being produced? They are produced via interaction of 2 different proteins. One of the proteins is an acyl carrier protein and another protein is an enzyme. But there is a problem. There are many different types of acyl carrier proteins in the cell, but the enzyme needs to interact with the correct acyl carrier protein to generate a specific signal. How it does that? We do not know that. We are trying to understand that in our lab. If I provide you an analogy, if I have 6 different colored papers and I have a blue scissor, the blue scissor needs to pick only the blue paper to create this beautiful blue flower, which is a message of love. How does the blue scissor only picks the blue paper? We do not know that. If I provide you another analogy, if a postman needs to drop a specific letter to a specific mailbox, he must have some prior information to do that. What is the information? We do not know that. If we know the information, then we will be able to seal the scissor and the mailbox shut. As a result, there will be no production of chemical messages, and there will be no effective communication among bacteria. As a result, the initiation of an infectious attack by bacteria will be prevented. Ultimately, we will be able to treat bacterial infection without using antibiotics. Thank you.
Abu, why don't you step over here?
Thank you.
Abu, I think what defines a scholar for me is someone so excited about what they don't know. A lot of times when I don't know something, I get overwhelmed, but I love the energy that you possess in activating yourself toward what you don't know. So thank you for that. I also love the cartoon characters.
Thank you.
That was very well done.
Thank
you. You spend a lot of time, obviously dealing with something that, as some other scholars here today, at the microbial level, that there's immense power in something so small for our lives. What do you do to recreate after you've lived in that world for a while? Like, what do you do for yourself? And so you come back to it maybe a little more energized and refocused.
Yeah. So I always try to motivate myself. What is the primary goal behind when I first dived into this research and what dragged me in here. So I always try to keep it alive to pursue my interest. So like maybe some background reading, why this research is important and what would be the significant application once we have some information that helps me to.
Abu, let me clarify the question. I'm more interested in your bad habits, not just your good habits of excessive studying. So I want to know the things you do when you don't, let me rephrase the question. I was going to say what you do when you don't study, but you clearly are always studying. What do you do to take a break from study?
So Idaho is a great place for traveling. I go into different places, and whenever I get some time, I have a special interest for cricket. I play cricket.
Oh, fantastic.
And I hang around with friends. So
you know what? I think if you could explain the rules of cricket to me in 3 minutes, that would be a real accomplishment. Thank you, Abu, very much.
Thank you.
Thank you. Okay, next we have, our next participant is Christina Cowan. Christina, welcome to the stage. Oh, that's nice. Put the sign, it's what we call upstaging in the theater. Put the sign behind me and don't tell me. Well, clearly it's intermission, so enjoy it. Everyone, welcome back. So there are 4 awards possible that will be presented today. One of them is the Audience Choice Award, so we encourage you, the audience, to participate in that. There is a QR code on the paper here in the room and on your computer screens at home to engage with that voting process, and we sure hope that you will. So let's move on to our second half and the rest of our participants. Next we have Christina Cowan. Christina, welcome.
If the culminating goal of research is to improve the human condition, then our best efforts need to be spent on working with humans. Behind me is a picture depicting a cross-cultural training that local clinicians received in music therapy. During this training, I had the chance to observe the mental health benefits of African drumming. Through that experience, I understood that with the arrival of nearly 500 Boise refugees this year, there is a need not only for these types of trainings, but they can become integral parts of the Boise community. I've been taught through my clinical training that working with refugees can become a very difficult barrier in local clinical staff. They're not receiving the correct, adequate training in acculturation or in experience assisting the refugee population. Considering this fact, I wanted to look into the study of what are local Boise clinicians doing to support the refugee population. So I want to tell a story. After experiencing horrific and abusive conditions, Lucy, a Boise refugee, resettled in America with 6 months to establish a self-sustainable lifestyle. During this time, she had to acquire housing, establish a job, and learn the language, all without any support from her friends, family, or culture. As Lucy finally gained personal physical safety, she only had started her battle with depression. This was due to Boise's loss of cultural identity and to symptoms related to complex trauma from experiences overseas. Recent research indicates that people like Boise are pretty typical. Refugees experience losses like this all the time. Considering this fact, the aim of my study was to identify what local clinicians needed in order to adequately serve this population. So I took a sample of, and I'm taking a sample of local clinicians, local clinical staff, through qualitative data to determine what they need. What we found and what we are discovering is that current evidence-based practices and interventions are not assisting them. So we need to make a change. If we can make a change and assist these people in getting the cultural integration that they need in their lives, we can help these refugees experience a more successful life overall. I believe this study is important because refugees are finally at a place where they need the assistance to have their voices heard. Thank you.
Hi, Christina.
Hi,
How are you doing?
I'm good.
That was great. You did great.
Thank you.
So you're not looking through a microscope. You're looking at our community and trying to discover what's happening.
Yes.
It feels like, from a global perspective, you know, the idea of the refugee is constantly moving, that in some ways we're all refugees in some ways. We're able to travel the globe
Absolutely.
like we were never able to before.
Yes.
How do communities make room and adapt and redefine themselves?
Absolutely, yes.
Yeah. Well, thank you for that. It was wonderful. What are you doing to recreate yourself?
I didn't expect that question. Let's see, I like to read.
Then you need more recreation. If you did not expect it. You like to read? What do you like to read?
Everything. There's a long list. I like to read stuff that's related to social work, which can become kind of like work, but I find it fascinating.
We already dealt with this. You can't.
We already dealt with
homework.
that.
You can't. Homework can't be on the list. What's the guilty reading?
Oh, fantasy books for sure.
Really?
Yeah.
Yeah. Okay.
Definitely.
Good. And what else? What do you like? What do you do out in the community? That's restorative?
Well, I have 4 kids.
Oh, okay.
So,
yeah,
they get me out
Yeah.
and get me involved in the community, and we love to go to the Greenbelt. We love to go down to the zoo. We try to, get their energy out. We go everywhere.
That's right. Get them tired out.
Yes.
Well, Christina, thank you so much. Well done.
Thank you.
I appreciate it.
I appreciate it.
Thank you, Christina. So next, our next participant is Jacob Manzy. Jacob.
Our electronics are constantly improving, changing, and sometimes in need of repair. What do you do when your electronics break down, when your phone needs a new antenna, battery, or screen? You probably take it to the Geek Squad or the Apple Genius Bar, right? Or maybe the more adventurous or tech-savvy among us take a trip to Radio Shack or order the parts you need to do a DIY repair or upgrade. Now, picture us as a civilization living 50 years in the future, on the moon, Mars, or somewhere else. There's no Radio Shack or Best Buy to make sure that your electronics are in proper working order. Plus, it costs about $10,000 per pound sent into space, so I don't think Amazon Prime is going to ship to you either. My research is in developing a novel technology called plasma jet printing. Uniquely, this printer uses a small jet of plasma to spray nanomaterials onto a substrate and create what we call a printed electronic. In the figure on the left, you can see the black flecks are the nanomaterial being passed down through the purple plasma and onto the yellow, flexible substrate below. The inset shows a figure of one of the plasma jet printers that we built here on campus, where you can clearly see the purple plasma jet going out to the side. Now, there are other technologies that can print materials, such as inkjet or aerosol jet printing, but they all require a post-processing step that usually involves high heat or intense UV light. These steps are costly, but they're crucial for the functionality of the printed electronics. They also limit what substrates can be printed on, as they need to be able to withstand the high heat or intense UV light. Because of the plasma in my setup, there's no need for post-processing, as the materials come out of the printer fully functional and ready to use. The plasma also doesn't burn or melt the substrate, so even paper and thin plastic can be printed on where they couldn't before. The plasma also makes it so that the printing is independent of gravity, and a group recently showed on a zero-G flight that the plasma jet printer can work in space and microgravity environments and can even print in any direction. So far during my research at Boise State, I've printed parts for a thermoelectric generator, a device that converts thermal energy into electricity. Think of it as something that might charge your Apple Watch using the heat of your body. I've also printed flexible and conductive gold and silver wires, and I'm working toward printing a fully functional antenna, as well as strain and optical sensing devices. This research is contributing to the ability for multiple materials to be printed onto one flexible functional circuit like the one this astronaut is holding in the figure on the right. Thankfully, this technology won't be for a civilization 50 years in the future, but functional in the next several years for both astronauts and civilians to print antennas, batteries, biosensors for health, solar cells, and more. Thank you.
Okay, so let me get this straight. You're going to print technology.
Yes.
And they'll have the printer to do that up there, right?
Yes.
All right.
Very cool.
I'm not sure I believe you, Jacob, but I don't know any better, so maybe I will. That was fantastic.
Thank
wow,
you.
It's like science fiction, but not fiction.
Yeah, it's really cool.
Yeah. What was the problem that got you started down that path? What were you trying to solve?
There's a lot of things that break in space, or you need to come up with something like that you just didn't have, and it takes a long time to send something up into space.
Yeah.
So
Was there something? Let's go back further, Jacob. Let's go back into your childhood. What in your childhood might have led you to this area of research?
I'm just very interested in solving problems, and I love applying my mind to science.
Right.
So kind of just.
And you even
myself
did as
in
a
this.
kid?
Yeah. Yeah, I grew up on a farm. And often we would build things or invent new games or whatever we wanted to do. Well.
See, I knew we would get there. All right, good. What do you do for fun?
Right now, snowboard a lot.
Just right now, like you just have discovered it, or you've always done it?
Well, I've only done it for a couple of years, but during the winter, it's like, oh, what do you want to do? Let's go snowboarding.
Yeah. Do you ever have any scientific breakthroughs on the hill? Gravity, I'm sure you've discovered gravity.
Yeah. Trying to think. No, mostly just snowboarding is a time to take a break from
Getting away from it.
Yeah.
Well,
science.
excellent. Thank you so much, Jacob. Appreciate it.
Thank
well
you so
done.
much.
Yeah.
Thank you.
These kids are amazing. Our next kid, our next participant is Jessica Barnerton. Welcome.
Microbes are bacteria and fungi that are virtually invisible. And despite their small size, they're on and in almost all living things. The natural world's teeming with microbes. In fact, the average person is carrying around between 2 and 6 pounds of microbes with them every day. But don't worry, the majority of those microbes live in harmony with their host and play a vital role in human and ecosystem health. And even though microbes are very small, they play complex relationships in the communities they live in. Now, a microbial community isn't too different from a human community. If you think of your favorite ski town as a community, each resident does a different job. Barista, gear rental guy, food service, but together the community's job or function is the ski town. Microbes are exactly the same. Groups of microbes can work together to do a job, and that job can change depending on the resources or the environment. Now, microbes are just one member of even larger communities. Like the lake pictured here, the microbes are interacting with the plants and the animals and the insects, with each member doing an important job. My research works to understand how microbial communities perform and how those jobs might change. But conducting an experiment on a 1,000-acre lake is really hard. I need to be able to measure and manipulate the microbial communities. So in essence, I need a miniature version of an ecosystem. And that's where pitcher plants come in. Each leaf on a pitcher plant is a cup that captures rainwater and hosts a diversity of organisms from insects, arachnids, and microbes. Imagine a whole ecosystem that you can hold in your hand. I use pitcher plants and the microbial communities that are associated with their leaves to test hypotheses about microbial community function. So, why do we care about the teeny tiny microbes inside a random carnivorous plant? Well, I studied the pitcher plant system, but you too are a standalone ecosystem. And all the microbes that live in you are directly related to your health. Just like the microbes in a pitcher plant are directly related to their health, and how the microbes in that lake are related to the health of the entire aquatic ecosystem. I'm interested in understanding how communities respond to a changing world. From fires and droughts to floods and storms, changes becoming more frequent and more extreme. So my research works to understand how communities respond to these changes with the hope that understanding these processes on a small scale can help to protect our planet's diversity on the large scale. Thank you.
Just become aware of my microbial life.
Don't worry, they're helping you.
All right, good. I'm going to take your word for it, but the fact that they have a they now starts to make me wonder, but good. Jessica Barnerton, sorry, I mispronounced your name, my apologies. What was the first thing that opened your eye to this idea of a microbial community? What was the first thing that you said, that's what I'm interested in? What took you there?
I've always been interested in plants, and when I did my master's research, I learned about the diversity of fungi that live on plants. Mm-hmm. And so I've slowly just transitioned to understanding more about the fungi and the bacteria and the communities they form. And so I think it's pretty impressive how important they are to all kinds of systems.
Yeah, and do you remember the first time you looked through a microscope and saw like this whole new world?
When I was little, my mom bought me a microscope at a garage sale, and I set it up in my closet and looked at everything I could look at underneath it.
Fabulous.
Yeah.
That's fabulous. What do you do when you're not looking at the microscope?
My family and I like to go and explore Idaho, do outdoorsy things like camping and hiking. And things like that. Yeah.
When you're out there camping with your family, do you lecture them? Do you give them the lowdown on the microbial world?
I try not to go too crazy, but my daughter loves fungi and moss, and so she's a little explorer on her own, yeah.
The legend continues.
Yeah.
Well, thank you so much. We appreciate it. Well done. Okay, our next participant is Rebecca Miller. Rebecca.
Imagine the next time you go on your weekly grocery store run, trying to purchase everything you need without getting anything packaged in plastic. That would be pretty hard to do, and that's because plastic packaging has become essential for the protection and preservation of consumer goods. In fact, while a lot of plastic packaging may seem fairly simplistic or even be very thin, plastic packaging can be very complex, containing multiple layers in order to meet various properties that cannot be met with a single component material. For instance, plastic packaging is resistant to heat, UV light, and moisture, all while being durable, flexible, lightweight, and inexpensive. This material complexity leads to positive things, such as a longevity of shelf life. However, it makes the recyclability of these materials non-existent, and that's because currently there is not a feasible separation system in place for the various layers. Likewise, every year there are over 8 million metric tons of plastic packaging alone that gets discarded as unrecyclable waste, contributing to the plastic waste accumulation that we're experiencing in our landfills and environment. To combat this, my research focuses on making these types of materials recyclable through utilizing degradable polymers. More specifically, I design new types of degradable polymers that can come apart on command when exposed to a specific trigger. Implementing these polymers at the interfaces of the various layers can enable their separation and therefore increase recyclability. Another way to think about it is that current multi-layer packaging is like cooking an egg on a pan without any oil. The egg cooks fine, but there really is no simple, easy, clean way to remove that egg from the pan. Adding degradable polymers at the interfaces is like adding oil to the pan. The egg cooks fine, but now it's easily removed when desired. Adding the degradable polymers to this design will not inhibit the material's original function, but will allow clean separation when needed. What's particularly exciting about my work is that I can selectively tune these polymers to come apart in select environments, meaning that I can align material separation to occur with processes already in place in industry, making this easily accessible and applicable on a large scale right away. With my work, we can continue to reap the benefits that plastic packaging offers while limiting the amount of plastic waste that we contribute to. Thank you.
It sounds almost too good to be true.
Right.
But if it could help me get my recyclables to the bin in a smaller, that would be fantastic. So I vote yes. I vote yes for it.
Awesome.
What took you to this topic?
To packaging.
Like, what attracted you to this area of research?
I think just having that constant motivation of like, this is an issue that needs to be solving. And it's so important throughout the Ph.D. to have a constant like motivating factor that's pushing you to push
You've been hearing about global warming most of your life, I would imagine, right? Mm-hmm. As I've asked some of the other participants, was there something in your childhood that maybe set a course correction to this place you are now in research?
I've just been always very interested in science. My mother was a high school chemistry teacher, and that sparked an interest at a very young age.
Fabulous. Did you ever go into her classroom when you were little?
When I was little, yes.
I went into my mother's classroom when I was very little, and it really was an impact on me.
Definitely.
Well, thank you so much. That was well done.
Thank you.
Thank you, Rebecca. So next, our participant is Dalton Miller. Welcome, Dalton.
Thank you, guys. I appreciate it. All right, we'll go ahead and get started. So, chronic wounds, seen here on your left, are wounds that fail to heal for months to years from their initial onset. They affect more than 6.5 million Americans every single year, and their treatment costs northwards of $50 billion annually. Now, obviously, that's a very large sum of money. That's very expensive. But what's significantly more costly than that is the amount of pain and suffering that people with chronic wounds have to go through. You see, once a week, these patients will go into a wound care facility where a doctor will unwrap their wound, snag a bottle of antiseptic, and vigorously scrub out the wound site. Now, this is to kill any bacteria or fungi that might be present that could cause an infection. But also, as you might imagine, scrubbing an open wound is an incredibly painful procedure. Now, after this, the doctor will trim any dead tissue from the wound site, rinse the wound out, wrap it back up, give the patient a pat on the back and say, well, we'll see you in a week. Hopefully that will heal. Of course, the doctor can't actually say when or even if the wound will heal. I don't think that any of you would reasonably describe that procedure as ideal, effective, or even particularly good. And it's to that end that we're working on developing a new method for treating chronic wounds. It uses an ionized gas, also called a plasma, which you can see here on your right, and it works by generating highly reactive molecules that serve two different functions. Now, the first of those functions is sterilization. The molecules are capable of killing bacteria and fungi in the wound site, and because we're using a gas rather than a liquid, no vigorous scrubbing is necessary. Of course, the scrubbing is the worst part. The secondary function that these serve is in wound healing signaling. To just use an example, if you get a cut on your hand, your body will produce these exact same molecules to signal to the cells around the wound to begin healing the wound. By directly introducing these molecules to the wound site, we can increase the patient's natural rate of wound healing and speed up the wound healing process. Now, right now, we're only testing in pig wounds. We want to test for safety and efficacy before moving on to human trials. But what we're seeing so far is reassuring and very encouraging. With only a 10-minute treatment, which is shorter than the amount of time it takes to scrub out the wound, we're seeing good sterilization and no damage to the cells of the pig wound, which is fairly encouraging. Now, we hope that eventually this will be used clinically, both to decrease the amount of pain that people with chronic wounds have to go through, as well as, more importantly, increasing wound healing rates so that these patients can have their lives back. Thank you. All right. You got a recreation pun for me? I'm ready.
I should do something different then.
You should.
But I'm not prepared.
No, you're fine.
So this is obviously important work. Thank you so much. I know I took a parent to the wound center, and I know what you're talking about. It is painful in many, many ways. So thank you for the work.
Well, thank you.
Was there something in your life that drew your attention to this, or?
I wish I could say yes. I wish I had a really good story. Okay. But I actually stumbled into this research entirely accidentally.
Yeah.
Originally, I wanted to go to medical school, and a requirement for medical school is doing research.
Right.
And when I was, when was it? The summer between my freshman and sophomore years of undergrad, I got an email that said, hey, there's this research thing, you should sign up for this research thing. And so I signed up for the research thing. And then I stayed on for forever, and they can't get rid of me now because I absolutely fell in love with it.
Oh,
so,
that's fabulous.
Yeah, and that's,
well, thank you. Thank you for the work. So, all right, so then what, are you ready to answer this question?
I am.
All right. What do you do to recreate?
It's a good question. So I would say for me, probably reading and movies are probably my favorite things.
I'm going to take both those off the list.
Okay. Going to do something else?
Yeah, it's got to be physical in nature.
Physical in nature. Okay, it's going to sound kind of weird, but I like to go on like just long walks and just sort of think about things.
Nice. Well,
refreshing.
A lot of great artists and scientists did that, right? Do you do you get often breakthroughs in when you're engaged in those walks?
It's sort of like halfway. So for the first half, I'm kind of stressed and I'm just kind of decompressing a little bit.
Yeah.
And about halfway through, my brain is more creative and I think of things that I maybe hadn't before. Wonderful. So I have like 8 more things that I want to do with the device that we're not doing yet.
Well, if I ever see you on a walk, Dalton, I will not disturb you because I know important work is being done.
No, you should say hi. I like talking to people.
Okay, good. Well, thank you very much. That was well done.
Thank you.
Bravo.
And thank you all as well.
Here's where we say last, but certainly not least. Our last participant of the morning is Danielle Siegel. Welcome, Danielle.
There's one thing that I can guarantee that every single person here today has in common, and that is that at one point in time, each of us was a baby. You would think that since this is something that we all share, that the research would be exhaustive, and we would know everything we possibly could about how babies develop and what products keep them safe. Surprisingly, this isn't the case. But the field of baby biomechanics is looking to change that. So then, what is baby biomechanics? Well, it's essentially the study of babies in motion And for me, I specifically look at how babies achieve a roll from their backs to their tummies. For most adults, this is a simple task that doesn't require much thought. But for babies, it's an important developmental milestone. To initiate and complete a roll, babies must use their whole body. And before they learn to crawl or walk, rolling is their main way of interacting with their environment in daily life. Currently, only a handful of research papers have touched on the topic of how babies achieve a roll. One in particular, by Kobayashi, that was published in 2016, found that there was actually 6 different coordinated movements that a baby may use. And although these 6 different coordinated movements have been found, no one has explored what muscles babies use to achieve a roll. This is the new and innovative part of my study. I was able to have 30 healthy babies between the ages of 3 and 7 months participate in my IRB-approved study. I had the babies lie flat on their backs on a playmat for 5 minutes or until they achieved a roll. A GoPro camera recorded their rolls to determine the coordinated movement type they used, and electromyography, or EMG sensors, were used to record the muscle activation of various muscles. This tells us what muscles are being used more and what muscles are being used less for the different coordinated movements. And so far, this has actually been shown to show us what muscles babies use to achieve a roll and has shown significant differences between the different coordinated movements. Having a strong and complete understanding of how babies achieve a roll is critical to keeping babies safe and advancing the knowledge in healthcare and engineering. If we have a better understanding of how healthy babies are developing, we can then understand when a baby's development may not be as expected, which could indicate other health concerns. This is crucial information for pediatricians and for families who may not have direct access to healthcare. From home, parents would be able to monitor their baby's rolling to ensure that their development was on track. This research could also lead to improved regulations for infant safety when designing new products. If we have a better understanding of how infants achieve a roll, we can better understand the key product design features that are needed to keep babies safe. Ultimately, babies are our future, and advancing the field of baby biomechanics is pivotal in keeping them safe as technology grows. Thank you.
Danielle, I'm trying to think if I could put together 6 coordinated moves, and I haven't come up with a list yet, so this is fantastic.
Yeah.
Was this fun research for you?
Yeah, it was great. I mean, I get to play with babies.
Yeah, I was
so
gonna say,
like,
that would
it's
be
pretty
so
fantastic.
great. And they were cooperative with you? They didn't ask for breaks or, you know, a higher wage or call my agent or anything like that?
I mean, they have their ways. They cry a lot, but
Sure.
we have to work around it.
Fascinating.
Yeah.
So what do you do to recreate, Danielle?
Like for fun and
For
what?
Yes, for fun.
Okay.
Fun's allowed, you know.
Fun's allowed. Okay.
Yeah.
Well, I'm in grad school, so I don't know. Like, there's not a lot of time.
It will be allowed very soon.
Okay. So, well, I have always been a dancer growing up. So,
Is that right?
Yeah. So I love still being involved.
Are you at all in the dance program here at Boise State? Like, do you know Marla Hansen?
Yeah, I was when I was in undergrad. I
Fabulous.
took classes from her and stuff.
Yeah,
so.
she's a wonderful colleague. We love her.
Yeah.
And is there a particular type of dance that you especially love?
Probably more on the jazz side, less
Yeah.
on the ballet side.
Do you have that experience of kind of losing yourself when you dance? Or
Yeah,
one of the-
absolutely.
So you're a good dancer.
Yeah, different world. Definitely takes away from the stress. So.
Fabulous.
Yeah.
Well, Danielle, thank you so much. Wonderful
Thank
work.
you.
Okay. Okay, well, thank you so much. And please, a round of applause and acknowledgement to all of our participants. Bravo. Yeah, it's an amazing task. And so now I'm going to invite the judges to engage in their amazing task and allow them to leave the room and deliberate. The results of the contest today will be announced on Facebook Live, the Graduate College at Boise State, a Facebook Live announcement at 1 o'clock today. A couple other things, a reminder about the Audience Choice Awards. Pay attention to that Q code. It'll allow you, that QR code, which will allow you